Hi everyone, welcome to Chapter 4, Cell Structure. This is the first lecture in a two-part series. So again, like most of this stuff up to this point, this is not necessarily new information for most of you, just kind of a review. You understand that the cell is the basic building block of all life. And so whether it's a bacteria that causes tooth decay or a mushroom that springs up in your yard after a warm summer shower, the little bat that pollinates the saguaro cactus in the Arizona desert, and yes, even the cactus, everything is made up of cells, and that includes us humans. So in this chapter we'll examine the structure of cells, locate and identify various components common to all cells, and we'll learn how cells work together to build greater structures like tissues, organ systems, and ultimately organisms. So our objectives for this chapter differentiate the two basic cell types, describe the basic structure of the eukaryotic cell, describe the basic structure of the prokaryotic cell, describe the structure of the cell membrane and explain its importance to the survival of not just the cell but the organism as well. Locate and identify important cellular organelles. Describe the structure of cytoskeleton and explain its importance, and briefly explain how cells communicate with one another. So it never hurts in situations like this to at least appreciate the historical significance of what we're talking about, in this case cells and ultimately cell theory. So Robert Hooke was an English scientist back in the 18th century, well 17th century and early 18th century, was the first person to describe cells. Here is a little sample or a little sketch of his sample of cork cells. He thought that these, he took a little slice of cork and looked at it under his fancy microscope there, and he thought that these little units looked like the rooms in a monastery or the cells in a prison, and so he called them cells. A contemporary of his, Antoine van Leeuwenhoek, was Dutch, and there's his little microscope there, little handheld ditty, and he looked at samples of water, drops of water, and in those samples of water he found all of these little organisms swimming around. He made sketches of them, as you can see here, and he called them animalcules because they were swimming around, moving around like animals, because plants don't move, right? So they were swimming around, so he likened them to little tiny animals. Animalcules was his term there. Matthias Schleiden in Germany in the 19th century made the proclamation that all plants are made of cells, and Theodor Schwann, another German, declared that all animal tissues are made of cells. And from those proclamations, and for that information, we come to the cell theory, and this is a sort of the current, this is the new cell theory, okay? The revised cell theory. This is good for you to know. All living things are composed of cells. Cells are responsible for all the functions of an organism, including metabolism, heredity. Cell is the basic building block of all life, and cells arise from pre-existing cells. Cells do not just appear out of thin air. They do not just spontaneously appear, right? They come from pre-existing cells. Okay, cell size is limited. Most cells are relatively small due to reliance on diffusion of substances in and out of the cells. We'll talk about some mechanisms of getting different substances into and out of the cell with active transport and transmembrane proteins and that sort of thing. But for the most part, cells rely on simple diffusion to get things in and out. And that rate of diffusion then, how fast it goes or how slow it goes, is affected by surface area, temperature, concentration gradient, and the distance the material has to travel once it gets inside of the cell to get to the place where it needs to be to do whatever it needs to do. Temperature, as you warm things up, the rate of diffusion increases, right? So the faster diffusion takes place as things get warmer and warmer and warmer. Concentration gradient simply refers to the amount of a substance in a particular area. So if you have a high amount of whatever substance it is on one side of the cell membrane, for instance, and a very small amount or zero amount of that same substance on the other side of the membrane, say on the inside of the cell, and that cell needs that stuff on the outside, then because of the difference in gradient, because of the difference in concentration, we call that the concentration gradient, that stuff on the outside will diffuse very quickly across to the inside. We'll talk about surface area here in a second. So organisms are made of many cells, or organisms rather that are made of many cells have an advantage over an organism composed of very few large cells. So the more and smaller cells that an organism has, it has a much greater advantage to life over an organism that is composed of very few or limited amount of really big cells. And that has to do with surface area to volume ratio. So as a cell's size increases, as a cell gets bigger, its volume increases, of course, much more rapidly than its surface area. And we'll look at that in a second with a diagram. That's called the surface area to volume ratio. And we'll see how that affects the rate of diffusion for a particular cell. So here we see in this illustration, here we see a cell with a radius of 1 unit, whatever the unit is, a micrometer, an inch, it doesn't matter. You pick the unit, whatever the unit is, but it has a radius of 1. And we're going to compare that to a cell that has a radius of 10. 10 times larger is this cell than that one. So if we look at the surface area of this little tiny cell, one unit, radius of 1, 12.57 square units. If we look at the volume, okay, 4.18 cubic units. And then if we look at the ratio of surface area to volume, we get 3 for this little tiny cell. Okay. You don't have to remember numbers. You don't have to do math here. I just want you to appreciate what's going on as we go from a radius of 1 to a radius of 10. 10 times larger is this cell. And you might think that the larger the cell, the better it is for life, or the more beneficial are large cells to an organism's existence. So if we raise the radius 10 times, look what happens to the surface area. The surface area definitely increases to 1257 square units. The volume increases as well dramatically, 4189 cubic units, but when you compare the surface area and volume via the surface area to volume ratio, look what happens. It drops exponentially. It drops dramatically to 0.3. so that a small cell with a small radius of 1 has a larger surface area to volume ratio than does a large cell that is 10 times bigger, which only has a surface area to volume ratio of 0.3. Okay, so what does it all mean? That means that diffusion across this cell will occur much faster because of this large surface area to volume ratio than diffusion across this cell or into this cell or across the cell membrane into this cell that is much larger because of its much smaller surface area to volume ratio. So the point here is the larger the cell, the smaller the surface area to volume ratio, and in turn the slower diffusion takes place across this into the cell, into or out of the cell. So think about this. What if you had some necessary nutrient that needed to get into the cytoplasm of the cell? Right? Because of the large surface area to volume ratio, diffusion is going to happen very quickly and get to where it needs to go, whatever the substance is, versus this large cell. Suppose you're diffusing down here and you need to go, I don't know, over here. Right? It's going to take much longer to get over there. Even if you came in from this direction, which is the shortest direction to there, it's still going to take much longer to go from there to there than there to there. You can't even see that spot in this little tiny cell. All right, so the point here is diffusion takes much longer the larger the cell is. And that's why it's to the advantage of any organism to have many more small cells than it is to have a few large cells. It seems counterintuitive, but it's definitely not when you compare the surface area to volume ratios of these two. Okay, so let's talk about cells and cell size. So here's a little ruler down here, a little measuring stick, if you will. And it says relative sizes of cells on a logarithmic scale, okay? So this little mu m, that stands for micrometer or micrometer. mm, I think you'll understand, stands for millimeter. 1m stands for meter. And just for reference sake, okay, if you have 1 meter, right, 1 meter is a yardstick and 6 inches, so it's 3.5 feet. A millimeter is 1,000th of a meter, so in a yardstick plus 6 inches, in a meter stick, you have 1,000 millimeters. Micrometer stands for 1 million, or it stands for millionth of a meter. So in 1 meter, 3.5 feet, you have 1 million micrometers. And this way down here says nm, that's nanometer. So that's 1 billionth of a meter. So way down here, these things are 1 billionth, or in this case, it says 2 nanometers. Now that means 1 billionth of a meter. These things are that size. All right, so really, really tiny down here, really, really big up here. Here we see an adult. If we measure using a meter stick, 1 meter, 3.5 feet, that's, I mean, on a large female, maybe that's 6 feet. 5 feet, all right, 1.5 meters perhaps, okay? So that's how that works. Now you see that it says naked eye. So anything from here 2 there you can see with the naked eye, with the largest cell in nature being the ostrich egg. All right, next to that in this particular diagram is the chicken egg, right? Chicken egg, that's an ovum, an unfertilized chicken egg that you pull out of the refrigerator. That's an ovum, right? That's an egg that hasn't been fertilized. That's a pretty large egg. You certainly can see that with the naked eye. Look at the frog egg. Frog eggs are easy to see. You see them in bunches in vernal ponds during the spring and throughout the summer. They're easy to see. They're pretty large. A couple millimeters here. Here's the human egg. Human egg is still visible with the naked eye. You don't need a microscope to see a human egg. It's small, but it's still a relatively large cell. So it's fairly easy to see. Now when you get down to plant cells, a typical plant cell and animal cell, these are microscopic. All right, but you can see them. Here it says light microscope. So you can see them easy with a light microscope. I'll show you some examples. Right, very easy with a light microscope. Now here, your bacteria and a mitochondrion right there. We'll talk about mitochondria. Those are little energy packages in cells. You can see them certainly with a light microscope. Now to see mitochondria, it has to be a really good light microscope, but it's very easy to see bacteria on high power with a light microscope. Roughly 1 micrometer in length. Now when you get down to certain viruses, in this case the flu virus, it's just beyond visible with the simple light microscope, but you certainly can see it with an electron microscope. And other viruses, Ebola virus is pretty big, so you can see that with a light microscope, but viruses like coronavirus and flu virus and the virus that causes the common cold, the rhinovirus, they're very easy to see with an electron microscope, but you can't see them with a light microscope. All right, here are lipids, as in phospholipid bilayer, around 1 to 2 to 3 nanometers. Very tiny, can only see those with an electron microscope. And then atoms themselves go beyond that, less than 1 nanometer, so very, very tiny. Okay, so this is just a comparison of different types of cells, and we will be sort of examining things in this range to this range. Okay. Okay, so we ended Module 1 with looking at the central dogma, and here it is again in review. And you know now that DNA, that genetic information travels in only one direction, from DNA to RNA to protein, and that process of going from double-stranded DNA to single-stranded RNA is called transcription, and we'll look at these in the next chapter in very much detail. And then going from RNA to protein, we call that translation. And yes, I know, even though this is the end of the process, this is not termination, but it's translation. Okay, so we go from DNA to RNA to protein. That's the flow of genetic information. That's the central dogma, very key pillar in modern biology. Okay, different cells have different features. However, all cells have some similar features. So all cells have a nucleoid or nucleus where DNA is located. Prokaryotic cells do not have a nucleus, but they do have a nucleoid, a central area where genetic material is located. All cells have a cytoplasm, that's the liquid portion that you find bound by the cell membrane. All cells have ribosomes, which synthesize proteins. Some cells more than others, but all cells have ribosomes. And all cells have a plasma membrane, which is made up of phospholipids. It's a phospholipid bilayer. It's 2 layers of phospholipids that we talked about last module. And so prokaryotes are things like bacteria, single-celled organisms like bacteria. Eukaryotes are, now, and these do not have a nucleus. These are going to have your nucleoid region. Bacteria have a circular DNA, a single chromosome, which is circular. Eukaryotes, EU stands for true, karyo pertains to the nucleus, so EU means true nucleus. So eukaryotes like plants and animals, and even things like, even though they're single-celled, amoeba and paramecia, they are eukaryotic. They also have nuclei. Organelles, lots of organelles, not just ribosomes, but endoplasmic reticula, nucleus, Golgi apparatus, all those things. And their DNA is linear, whereas prokaryotes have circular DNA. The features that are common to the two, cell membrane and ribosomes. So there are 2 cell types, as I already mentioned, and you know these. Prokaryotic cells have no nucleus. These include all of your bacteria. They are very small, range in size from about 0.5 to 5 micrometers. You can see them with a simple light microscope. Pro means early or before, karyo pertains to nucleus. So this is a cell that was generated before the nucleus, or this is a pre-nucleotic cell, right? Whereas eukaryotes, or eukaryotic cells, EU means true, karyo pertains to the nucleus. So these are cells with a true nucleus. They have a membrane-bound nucleus. These are large cells, roughly 10 to 100 micrometers, and they include all cells, all plant and animal cells. Okay, so here we see in this diagram a generic prokaryotic cell, a generic bacterial cell. All of these things that you see labeled you can find them on bacterial cells. Not all of these things exist on all bacteria, okay? This is just a generic example of a bacterial cell. Things to remember: prokaryotic cells do not have a nucleus. They do not have membrane-bound organelles. They do have a cell membrane, or another word for that is plasma membrane. I use them interchangeably. They have cytoplasm. That's the liquid portion contained within the cell. Bacteria have a cell wall. Here we see it in yellow there. That's like plants. Plants also have a cell wall, as we'll see. Bacteria have a capsule. Well, some bacteria have capsules. You can see it out there. That prevents them from, for instance, dehydrating in a very dry environment. All bacteria have ribosomes. That's these little things speckled in the cytoplasm. Plasmids, that's the circular DNA, the circular chromosome. Pili, some bacteria have pili. That's these little green little things sticking off here. That helps, pili helps some bacteria to stick on various surfaces, sort of like the Velcro thing. And some bacteria have flagella. That's this thing, this sort of tail back here that spins around like a propeller and allows some bacteria to propel themselves through the medium. Some bacteria have 2 flagella, some have 3, right? Some have flagella on both ends, so they can go back and forth, all right? Here's a typical eukaryotic cell, true nucleus. Again, all of these things are potential characteristics of eukaryotic cells. Not all eukaryotic cells have all of these things, right? So membrane-bound nucleus, there it is right there, nucleus. Membrane-bound organelles like mitochondria, endoplasmic reticulum, Golgi apparatus or Golgi body. All eukaryotic cells have a cell membrane, as do all prokaryotic cells. This is the cell membrane out here. All eukaryotic cells have ribosomes to make proteins and other things. All eukaryotic cells have DNA. We find that in the nucleolus or the nucleolus, which is found inside of the nucleus. Membrane-bound organelles, these are mitochondria, all right, also membrane-bound. And some eukaryotic cells have cell walls. That includes plants and fungi. And we'll look at a picture here in a second. So here's a bacterial cell in diagram and also under microscope. This is an electron microscope over here. This is a light microscope. You really don't see any details here. This is electron microscope where you can clearly see the nucleoid region. This is the genetic material. This is the DNA. See, it's a circular chromosome. See, you can follow the circle. Cytoplasm here, plasma membrane out there. Okay. This happens to be Escherichia coli, or you know it as E. coli. We hear about E. coli infections making a lot of people sick when meat is improperly packaged, processed and packaged, and that sort of thing. Here is a typical animal cell. Again, all of the things that could be present in an animal cell. Here's a light microscope view of some skin cells. You can see the plasma membrane, this sort of fuchsia line surrounding the cells. These are the nuclei in pink. And the dark area inside of the nucleus, you can clearly see the nuclear membrane, by the way. And the dark sort of spot, dark area inside of the nucleus, that's the nucleolus or the nucleolus. That's where, when a cell is not in the process of dividing, that's where all the genetic material is located, in the nucleolus. You'll notice that these cells do not have a uniform shape. Animal cells do not take on a uniform shape because they do not have a cell wall. Compare that to the generic plant cell. Now look at these cells. Again, simple light microscope here. Look at these cells. They vary slightly in size, but roughly all of these cells have the same shape. That's because of this rigid cell wall. The dot there in the center is the nucleus, and we don't see it here. Can kind of make it out in some of them. The central vacuole, which contains water, holds nutrients and that sort of thing. But this rigid shape of the cell is due to the rigid cell wall made up of, you know this, cellulose. And the plasma membrane lies just inside of the cell wall. There's little mitochondria. Here's nucleus, of course, nucleolus or nucleolus. These are chloroplasts. These green sort of oval things are chloroplasts, which only plant cells have versus animal cells. Animal cells do not have chloroplasts. That's where photosynthesis is conducted. There is a video that I want to post for you that you can look at, which shows you chloroplasts streaming around the central vacuole in plant cells. It's very, very cool, and it's really it's a great thing to see. When I do this class, the lab part live, my students are able to actually collect plant cells and look at them under the light microscope, and they can actually see the chloroplast streaming around. It's a very, very cool little exercise. All right. This chart just shows you the different types of structures in plant and or animal cells. Gives you their name, a little description, and their function. For instance, cell wall, made up of cellulose in plants, chitin in shellfish, lobsters, crabs, arthropods, insects, that type of thing. And it gives you the function. Cell walls protect and support. Here's the plasma membrane. Okay, regulates what passes in and out of cells. Nucleus is a control center of the cell. All right, so you can go through that chart on your own, just a little study aid for you, looking at the various structures inside of cells. And here's another little comparison chart of prokaryotic cells and then eukaryotic cells divided into animal and plant cells. You can see cell membrane, right? Present. Every cell has a cell membrane. Cell wall. Animal cells do not have cell walls, but plant cells do, and some bacteria do. What else we got here? You can go down through here. It's just, like I said, a little comparison. Here's mitochondria, absent in prokaryotic cells, present in both animal and plant cells. Okay, so that's just a little descriptive chart allowing you to compare prokaryotes like bacteria with plant and animal cells, and then plant and animal cells with each other. Okay, that is the end of this first part of chapter 4. In the next part, we'll look at the further structures of the cell, and we'll look at some organelles and go from there. So there you go. See you in the next part.